Hello, everyone. This is Rashi Gupta. In this podcast, we are going to discuss chimeric antigen receptor T cell therapy, otherwise known as CAR T cell therapy. CAR T cell therapy involves modifying a patient's own T cells to express a chimeric antigen receptor through genetic engineering. So far, it has been successful in treating several hematological cancers and has been approved by FDA as well. But much of its applications in solid tumors remain elusive. The idea for CAR T cells first emerged in the 1980s with the introduction of tumor infiltrating lymphocytes. These lymphocytes were extracted from the patients, expanded in a laboratory setting, and subsequently reintroduced into the patient's system. This approach led to complete remission in about 20% of individuals with metastatic melanoma, demonstrating the anti-tumor potential of T cells. Tumor-infiltrating lymphocytes naturally express T cell receptors that can identify tumor-specific antigens in a manner restricted by major histocompatibility complex, or MHC. This approach was further modified to bypass MHC restriction using immunoglobulin variable chain fragment to build chimeric receptors. Clinical trials for CAR T cell therapy were initiated in the early 2000s, primarily targeting hematological cancers such as leukemia and lymphoma. The basic production process of a CAR T cell requires integrating the single chain variable fragment from a monoclonal antibody with the signaling domain of T cell co-receptors. By designing different monoclonal antibodies targeting various tumor-associated antigens, the complementarity determining regions within the T cell receptors can be replaced, effectively bypassing MHC restriction. The single-chain variable fragment in CAR T cell plays a crucial role in tumor recognition by binding to tumor-specific antigens and transmitting activation signals to T cells. The chimeric antigen receptor structure is further stabilized with a transmembrane domain and a spacer region, typically derived from immunoglobulins or CD8 alpha, ensuring a strong connection between extracellular and intracellular components. The choice of the initial T cell population significantly impacts CAR T cell manufacturing and final product quality. Autologous CAR T production typically begins with mature T cells, either isolated or within the peripheral blood mononuclear cell population. These cells differentiate into various subtypes, including naive T cells, stem cell memory T cells, central memory T cells, effector memory T cells, etc. The differentiation state of the selected T cell population correlates with anti-tumor efficacy. Upon antigen stimulation, T cells expand clonally, with some differentiating into short-lived but potent effector cells, while others form long-lived memory T cells that provide a sustained immune response. Compared to terminally differentiated effector T cells, naive or memory cells exhibit greater longevity and a reduced risk of acute toxicities. These findings support the enrichment of naive and memory T cells in manufacturing process. After selection of the T cell population, activation of the intracellular domains containing tyrosine-based activation motifs from the CD3 immunoreceptor are triggered via cytokines like interleukin-2, interleukin-7, or interleukin-15. This leads to proliferation of targeted cells that can now be isolated by leukapheresis. Leukapheresis is a specialized blood filtration process that separates and extracts white blood cells while returning red blood cells, plasma, and platelets to the patient. This procedure minimizes blood loss and allows for the collection of a sufficient number of T cells for CAR T cell manufacturing. The CAR gene is introduced into T cells using viral vectors or non-viral methods such as CRISPR-Cas9, transposons, and mRNA transfection. While virus transduction via lentiviruses, retroviruses, etc., offers high integration efficiency, it carries risks such as insertional mutagenesis and replication competent viruses. Non-viral methods, particularly CRISPR-Cas9, allow precise genome editing by introducing a double-stranded DNA break at a target site, followed by homologous recombination for transgene insertion. Genetically modified T cells are cultured with anti-CD3 CD28 beads that eliminate non-T cells and promotes proliferation of selected transformed T cells via cytokine-mediated activation. Expanded CAR T cells can now undergo purification via pull-down assays before being stored for clinical use. All FDA-approved CAR T cell therapies undergo rigorous characterization, assessing safety, purity, potency, identity, and stability. The FDA has recently drafted standardized expectations for CAR T cell products to ensure consistency and safety. Since its discovery, CAR T cells have been developed with recurrent modifications for several generations. The first generation of CAR T cells contain only the CD3 signaling domain, resulting in limited effectiveness due to the absence of co-stimulatory signals. Full T cell activation requires both direct T cell receptor engagement and an additional co-stimulatory signal provided by molecules like CD28 or CD86 found on antigen-presenting cells. Second generation of CAR T cells were designed to enhance persistence and activation. Hence, they incorporated co-stimulatory domains such as CD28 or 41BB. While they show improved function compared to the first generation, challenges related to efficacy and safety persisted in certain cancer types. Third generation CAR T cells integrate multiple signaling and co-stimulatory domains, further strengthening T cell activation and persistence to improve therapeutic outcomes. Fourth generation CAR T cells, also known as T cells redirected for universal cytokine killing or TRUCs, not only express the receptor but also secrete cytokines like interleukin 12 to modify the tumor microenvironment. This generation includes two separate transgenes, allowing chimeric receptor expression alongside cytokine production to enhance anti-tumor activity. Fifth and the most recent generation of CAR T cells includes advanced modifications, such as cytokine receptors or transcription factors, to further enhance T cell function. It features an additional intracellular domain compared to earlier versions, often incorporating a truncated interleukin 2 receptor beta chain. This design provides greater control over CAR T cell activity, enabling drug dependent activation or depletion based on therapeutic needs. These improvements enhance precision and safety, making fifth generation CAR T cells more adaptable and effective in cancer treatments. Considering the vast number of cancer therapies available for patients, what stands out for CAR T cell therapy is its ability to replicate and persist in the patient's system like a living drug, leading to a prolonged anti-tumor effect. Compared to immunotherapies based on T cell receptors, CAR antibodies can recognize a wider range of tumor associated antigens independent of MHC restriction. Patients benefit from CAR T cell therapy as the treatment duration is relatively short. It requires only a single infusion, often followed by a maximum of 2 weeks of inpatient care. Additionally, since CAR T cell therapy does not involve intensive chemotherapy, patients typically experience a significantly faster recovery compared to those undergoing stem cell transplants, which rely on aggressive chemotherapy. Significant advancements have been made in treating specific hematological malignancies using CAR T cell therapy, particularly acute lymphoblastic leukemia, diffuse large B cell lymphoma, and other lymphomas. Some of the FDA-approved therapies include ABECMA for multiple myeloma, Tecartus and Lysocell for acute lymphoblastic leukemia, and Lysocell and Axicell for refractory large B cell lymphoma. Currently, no CAR T cell therapies have been FDA approved for solid tumors, though multiple clinical trials are underway. Solid tumors exhibit significant variation in antigen expression among their cancerous cells. Certain tumor associated antigens, such as carcinoembryonic antigen, are expressed during fetal development or in immune privileged regions. Because these antigens are primarily located within the cytoplasm, they remain inaccessible to chimeric antigen receptors, limiting their utility in CAR T cell therapy. CAR T cells may attack healthy tissues as well, expressing the target antigen, posing a risk to normal cells. Tumor antigens fall into two categories: tumor specific antigens, found only on tumor cells, allowing for targeted therapy with minimal side effects, and tumor associated antigens, present on both cancerous and normal cells. The tumor associated antigens poses a higher toxicity risk as CAR T cells can eliminate both tumor and non tumor cells, leading to off target effects. Another challenge for CAR T cells is suppressive tumor microenvironment. Higher expression of immune checkpoint proteins, regulatory T cells, and myeloid derived suppressor cells expressing inhibitory receptors such as LAG3 and CTLA4, cytokines such as TGF beta and IL-10, reactive oxygen species, tumor derived metabolites like lactate, adenosine, and L-arginine are responsible for T cell exhaustion and inactivation, reduced tumor killing capacity, weakened immune response, and poor prognosis. Myeloid derived suppressor cells, or MDSCs, not only suppress CAR T cell antitumor activity but also promote tumor angiogenesis and invasiveness by inhibiting T cell proliferation through arginase, TGF beta, and IL-10. MDSCs further contribute to immunosuppression by generating reactive oxygen species, which create an oxidative environment that disrupts T cell receptor signaling, downregulates CD3, and impairs CD8 T cells' function, thereby weakening antigen specific immune responses. Additionally, oxidative stress induces mutations in genes governing the cell cycle, including the tumor suppressor gene p53, promoting tumorigenesis and cancer progression. Barriers such as dense trauma also restricts T cell infiltration. This trauma consists of an extracellular matrix, blood and lymphatic vessels, immune cells, mesenchymal cells, and fibroblasts, forming a tightly woven network that hinders immune cell access. In some cases, the stroma becomes so compact that it renders the tumor nearly impenetrable by CAR T cells. The acidic environment inside tumor also promotes the formation of fibrous structures around tumors, restricting T cell motility and infiltration. A key metabolic distinction between tumor and normal cells is their reliance on aerobic glycolysis for energy production, even in the presence of oxygen, a phenomenon which is known as Warburg effect. However, this metabolic strategy is not exclusive to tumors. Effector T lymphocytes, essentially for anti-tumor immunity, also depend on glucose for expansion. This overlap results in nutrient competition, particularly in tumor regions with limited resources. Tumor cells actively export acidic byproducts, lowering the local pH to 6.8, which supports their growth while hindering normal cell function, as healthy cells require a pH of 7.3. This also poses as a challenge for CAR T cells. CAR T cell therapy can lead to acute toxicities like cytokine release syndrome. CRS initially presents with mild systematic symptoms like fever, hypotension, tachycardia, and hypoxemia. Though typically reversible if promptly managed, delays in diagnosis can occur due to limitations in severity grading criteria. CRS is linked to elevated levels of interleukin 6, interleukin 8, interleukin 10, interferon gamma, GM-CSF, macrophage inflammatory protein 1 beta, and monocyte chemoattractant protein 1. CAR T cell therapy also comes with economic challenges. Clinical trials for CAR T cell therapy outside the USA, Europe, and China are rare due to the high costs involved. Data from the EMBT registry highlight a stark contrast between this number and the vast population of oncology patients eligible for CAR T cell therapy, suggesting that approximately 99% of potential candidates did not receive the treatment. The primary barriers are the therapy's high cost, estimated at nearly $500,000, and the complex manufacturing process, which takes 21 to 35 days. Additionally, obtaining autologous T cells can be challenging in patients with advanced solid tumors, necessitating the use of T cells from healthy donors to address issues such as limited cell availability and compromised T cell quality. Additionally, studying CAR T cell manufacturing and its impact on therapeutic outcomes is inherently difficult due to the lack of control experiments and models. In autologous cell therapy, each T cell product is unique, with significant donor to donor variability, particularly in heavy pretreated patients who have undergone hematologic toxicity and lymphotoxic therapies. CAR T cell manufacturing protocols differ across trials and are often proprietary, making it challenging to directly link clinical outcomes to specific manufacturing process. Preclinical animal models serve as a crucial bridge between laboratory research and clinical application. Ideally, these models should accurately represent human disease, capture the dynamic pathophysiological changes during disease progression, and effectively assess potential toxicity. So far, such models have not been developed. To overcome these challenges, scientists are working on enhancing chimeric receptor design, changing targeting strategies, overcoming tumor suppressive environment, using combinational therapies, managing side effects, and improving accessibility to larger public. Advancements in CAR T cell design focus on improving specificity, persistence, and controlled activation. Strategies such as bicistronic CAR T, tandem CAR T, co administration, and co transduction aim to enhance CAR T cell proliferation, longevity, and functionality within the tumor microenvironment. To control the tumor microenvironment suppression, CAR T cells have been engineered to secrete cytokines that reshape the immune landscape. Drugs, as mentioned before, express cytokines like interleukin 2, interleukin 4, interleukin 8, interleukin 12, etc., to boost CAR T cell activation and persistence. CAR T cells engineered to secrete cytokines or therapeutic agents like IL-12, IL-15, and IL-21 to enhance anti-tumor activity are also called armored CAR T cells. Fifth generation or universal CARs improve flexibility and antigen recognition by separating the antigen targeting domain from the T cell signaling unit through a lock key system. This innovation enables CAR T cells to achieve nearly full antigen specificity. On-switch CARs further enhance control by segregating the signaling and co-regulatory domains. Small heterodimerized molecules assemble these components, allowing precise regulation of activation dose, timing, and location, thereby minimizing first pass and off tumor toxicities. For example, a CAR T cell system incorporating a folic acid fluorescent isothiocyanate switch exhibited selective cytotoxicity against folate receptor positive cells, while sparing folate receptor negative cells, improving safety. Natural killer cells possess receptors that differentiate tumor cells from healthy cells, and these receptors are incorporated into CAR designs to improve tumor recognition. Pooling CAR T cells with different specificities or engineering dual CAR T cells capable of recognizing multiple tumor associated antigens can also be done. To mitigate off target toxicity, suicide gene strategies such as inducible caspase 9 enable the selective apoptosis of CAR T cells that trigger adverse responses. Alternative approaches, such as CAR switches based on antibodies, expand targeting capabilities by incorporating a tag in a specific anti tumor associated antigen antibody. This allows redirection towards antigen expressing cells. Another innovative strategy involves remote controlled CARs, which remain inactive until activated by adapter molecules designed to modify targeting post therapy administration. Immune checkpoint inhibitors combined with CAR T cell therapy has also been proposed. Preclinical studies demonstrated that CAR T cell therapy combined with PD-CD1 blockade enhances synergy, achieving long-term survival without adverse effects. The CRISPR-Cas9 system has been employed to delete immune checkpoint genes such as PD-1 and CTLA4 to improve CAR T cell efficiency. CAR T cells can also be modified to express enzymes like heparinase and catalase to neutralize immunosuppressive factors. CAR T cells overexpressing heparinase can also facilitate tumor infiltration. Improving the homing ability of CAR T cells can enhance their therapeutic efficacy as well. CAR T cells can be preloaded into a biopolymer scaffold along with coactivating molecules. And once placed near the tumor mass, the scaffold gradually releases CAR T cells. Infection with oncolytic viruses can also further enhance CAR T cell infiltration, motility, and persistence by alleviating local immunosuppression. Recent clinical trials used microscopic titanium or nitinol plates loaded with CAR T cells, which, when implanted in the tumor site, significantly reduced tumor progression. Lipid based nanoparticles have also emerged as effective delivery systems due to their favorable physicochemical properties, offering advantages over viral vectors such as reduced toxicity, lower cost, modifiability, and no risk of genome integration. Researchers have also developed cell reservoir technology, which involves encapsulating CAR T cells, anti PD-L1 antibodies, modified platelets, and IL-15 loaded nanoparticles within the biocompatible hydrogel implant for generating a controlled release system for localized, sustained CAR T cell delivery. Additionally, modifying the tumor microenvironment, such as inducing interleukin 4 secretion, can shift it from immunosuppressive to activating, improving CAR T cell function. While autologous CAR T cell therapy remains a personalized approach, universal allogenic off the shelf CAR T cell therapies with low alloreactivity are under development. The primary source for allogenic CAR T cell manufacturing is peripheral blood mononuclear cells from healthy donors. Next generation manufacturing now enables final product formulation within 24 hours, drastically improving patients' access to treatment. One example is YTB323, a CD19 directed CAR T cell product developed using the T Charge platform, which requires less than 2 days of ex vivo culture. Reducing macrophages and eliminating monocytes in the tumor microenvironment can significantly lessen cytokine release syndrome severity. Inhibiting GM-CSF signaling can also help in mitigating CRS symptoms. A machine learning based method developed by Alex et al. Demonstrated over 90% accuracy in identifying cytokine release syndrome by analyzing cytokine peak concentrations. Traditional biochemical analysis and training blood morphologists to recognize CAR T cells are costly and time intensive. Deep learning, an advanced computer vision technique, has significantly improved medical cell image classification. A major AI challenge is optimizing target identification for CAR T cell therapies. Daniels et al. Addressed this by developing a CAR library containing over 2,300 synthetic co-stimulatory domains that influence T cell outcomes. Screening and sampling techniques have identified over 1,000 human tumor antigens cataloged in the Cancer Immunome database. The combination of CAR T cell therapy with various modalities such as chemotherapy, radiation, and immunotherapy has shown promising results. Following localized radiotherapy, cytotoxic T lymphocytes were drawn to irradiated tissue, triggering a localized immune response, while also exhibiting an ability to suppress tumors at distant sites, an effect known as abscopal phenomenon. Research indicates that radiation enhances the release of interferon gamma and damage associated molecular patterns, which in turn attract immune effector cells to the tumor microenvironment. This process improves the infiltration and transport capacity of these immune cells, fostering a more favorable environment for T cell activity. Moreover, localized radiation has been found to upregulate the expression of specific chemokines, including CXCL9, 10, and 16, which promote T cell recruitment and enhance immune cell infiltration within the tumor microenvironment. Additionally, studies suggest that irradiating tumors can increase the expression of major histocompatibility complex class 1 molecules and tumor specific antigens on tumor cells, further boosting immune recognition. Chemotherapy also plays a crucial role in modulating the immune response. Certain chemotherapeutic agents, such as doxorubicin, fluorouracil, gemcitabine, cyclophosphamide, and docetaxel selectively target and suppress immunosuppressive cells, including regulatory T cells and myeloid derived suppressor cells. Furthermore, taxane based drugs and Vinca alkaloids contribute to enhanced tumor cell recognition by promoting calreticulin exposure, leading to tumor cell destruction. Chemotherapeutic agents facilitate tumor antigen expression through several mechanisms. Autophagy induced by certain chemotherapeutic drugs trigger the release of ATP, which attracts dendritic cells and T lymphocytes, enabling deeper tumor infiltration. Dying tumor cells subjected to chemotherapy release damage associated molecular patterns, such as high mobility group box 1, which interact with toll like receptor 4, thereby promoting dendritic cell maturation and strengthening anti tumor T cell responses. Chemotherapy can stimulate tumor or stromal cells to produce endogenous type 1 interferon as well, while an increase in type 1 interferon further activates dendritic cells, facilitates cross priming of T cells, and enhances tumor suppression. Lastly, treatment with certain chemotherapeutic agents leads to the upregulation of mannose 6 phosphate receptors on tumor cells, making it easier for cytotoxic T lymphocytes released granzyme B to penetrate tumor cells and regulate their activity. Despite all the modifications, standardized procedures, models, and regulations are still needed to advance CAR T cell therapy. Most research and clinical studies are currently in phase 2 or 3, and much of its potential remains unclear. CAR T cell therapy has also been explored for autoimmune diseases, asthma, type 1 diabetes, HIV, hepatitis, cardiac conditions, and liver fibrosis. But these topics are outside the scope of today's episode. In this podcast, we covered what CAR T cells are, how they are manufactured, their benefits over other therapies, and the main challenges this emerging treatment faces. We also discussed some modifications and alternative strategies to address these challenges. While CAR T cell therapy is still in its early stages and not yet fully integrated into medical practice worldwide, it holds great promise for personalized cancer treatments. Thank you for listening.